This is a short video on the hematopoiesis flowchart, which is located in your lecture folder on blood. You might be looking at this and say to yourself, there's a lot more on here than what's in the flowchart. And you would be right. I've added additional information so that I can study hematopoiesis using only one 8x10 sheet of paper. Let's look at what I've done. The stem cell for all formed elements is the hemocytoblast. These are located in the red bone marrow. Recall that your formed elements include your erythrocytes. Recall that the formed elements include the erythrocytes, or your red blood cells, thrombocytes, or platelets, and your leukocytes. Examples of your leukocytes, recall, are your monocytes and your lymphocytes, which are agranulocytes, and your granulocytes, which include your neutrophils, eosinophils, and basophils. Recall from the lab packet that granulocytes end with "phil," and that'll make it easier for you to remember which of your leukocytes are granulocytes. The agranulocytes end with "cytes." Let's look at this chart. I've already filled in the stem cell, the hemocytoblast. Hemocytoblasts can differentiate into two different types of stem cells: myeloid stem cells and lymphoid stem cells. You'll need to know what cells arise from the myeloid stem cells and what cells come from the lymphoid stem cells. That'll be easy to remember. Think of lymphocyte when you see the lymphoid stem cell. The lymphoid stem cell gives rise to your lymphocytes. Let's begin with this side of the chart. I have the lymphoid stem cell giving rise to immature lymphocytes, which are called blast, so lymphoblast. There are two different types of lymphocytes which the lymphoblast can become, or two main categories: T lymphocytes and B lymphocytes. Recall that lymphocytes are part of your specific immune response or adaptive immune response. We'll look at what this means later when we study the immune system. But briefly, this means that these cells recognize only one antigen or foreign molecule and mount a response against that particular antigen. Let's look at T lymphocytes. For the functions of T lymphocytes, I've put down viruses. So T lymphocytes attack virus-infected cells or cells that have other types of intracellular pathogens, cancer cells or tumors, and grafts. Grafts are an example of foreign cells that you would get from an organ transplant. When B lymphocytes are activated, they begin to divide, and most of the daughter cells will be what we call plasma cells. Plasma cells release antibodies. Great, now I've done this side of the chart. Let's look at this other side of the chart, the myeloid stem cells. Recall, they're going to give rise to all of the other formed elements. So let's start with what we know. I've included pro-erythroblast in this box. Pro means before, so this cell I know will become an erythroblast, which is an immature red blood cell. There are different types of erythroblast, but the only two cells that arise from the pro-erythroblast that I want you to know are the reticulocytes and the mature erythrocytes. So let's talk about the reticulocytes. The reticulocytes are cells that leave the red bone marrow and go into the blood. They are anucleated, which means they have already ejected their nucleus, but they are not quite mature into erythrocytes. They will be in the blood for 1 to 2 days before they actually become erythrocytes, and their numbers are typically very low in the blood. If their numbers are elevated, that means that I have low oxygen carrying capacity in my blood. In other words, I don't have enough red blood cells. And increased levels of reticulocytes reflect increased erythropoiesis due to the low oxygen level or carrying capacity. So this has a clinical significance. If a patient has elevated levels of reticulocytes, then I know that they have increased erythropoiesis due to the fact that they're not carrying or delivering enough oxygen to their tissues. Once reticulocytes complete their maturation, they are erythrocytes. And the function of erythrocytes is to transport oxygen and carbon dioxide. Let's take a look at this box here. I've included this box because in this box, I have the term for red blood cell production, erythropoiesis. So anytime you see the term that poiesis, you know that you're producing or production of. The hormone that increases erythropoiesis is called erythropoietin or EPO, and it is released by hypoxic kidneys. So these are kidneys that are not receiving enough oxygen. You release EPO, and through a negative feedback mechanism, the EPO will go to the red bone marrow, stimulate the increased production of erythrocytes or red blood cells. That's going to increase the amount of oxygen that's being carried to the kidneys and other organs. And so you're going to return oxygen carrying capacity back to normal. So this gives me all of that information there. I've also included some other information. The hematocrit is the percentage of red blood cells to the total blood volume of your sample. So if you have a low hematocrit, that means you have low levels of red blood cells. Since you don't have enough red blood cells, your oxygen carrying capacity will drop. The conditions that result from a low hematocrit are called anemias. You could include another box that lists all the different types of anemias and their causes. You could also include another box that talks about high hematocrit levels and polycythemia. Go to your notes and figure out what polycythemia is and include it in this flowchart. Now let's look at thrombocytes or platelets. The myeloid stem cell will become a megakaryoblast, which will mature into a megakaryocyte. Megakaryo refers to the fact that these cells have a large nucleus. Pieces of the megakaryocyte will pinch off and become thrombocytes. So thrombocytes or platelets are not entire cells. They are fragments of these much larger cells called megakaryocytes. They contain granules that have chemicals which will be involved in blood clotting. The function, therefore, would be coagulation, which is clotting. Platelets are going to be involved in all of the phases of hemostasis. You might want to include some additional information here about that. Let's look at the monoblast. The monoblast will become monocytes. Remember, monocytes are agranular cytes. They are the largest of the leukocytes, and you learned about this in lab. When they leave the blood vessel, they become macrophages, which are large phagocytic cells. So they're going to be eating foreign cells and presenting some of the molecules of that foreign cell to lymphocytes, to T lymphocytes, to help activate them. So they're phagocytic. Their numbers increase also when you have chronic infections, such as during mononucleosis. Let's look at the granulocytes. We mentioned earlier that there are 3 types of granulocytes, and they all end with "phil": neutrophils, eosinophils, and basophils. For lab, you're going to need to know that neutrophils will attack bacteria and microscopic fungi. The levels of eosinophils will be elevated when you have parasitic worms. So they're going to be used to attack parasitic worms. they'll release digestive enzymes on the surface of the parasitic worm, and this will break down the worm. Their numbers are also elevated during allergies and asthma attacks. The last granular site that I have here are the basophils. Basophils store in their granules histamine and heparin. Histamine initiates inflammation, and heparin is an anticoagulant. In other words, it prevents blood clotting. So now I have all of this information that I have included. Let's look at this. With the thrombocytes or platelets, I've included an additional box that gives me the term of platelet production, and that is thrombopoiesis. The hormone for thrombopoiesis is thrombopoietin, released by the liver. Up here, I've included some additional information. Thrombocytopenia, which is an imbalance, is a low platelet blood count. Since platelets are required for blood clotting, that means I'm going to have decreased blood clotting. You should know the symptoms and what the role of the platelets are, or the roles in hemostasis. You can include that, add some additional information, what causes thrombocytopenia, different causes, and what is petechia, which I mentioned in the video. I've also included the term here for leukocyte production, leukopoiesis, and the chemicals that bring that about. And those chemicals or cytokines are your colony stimulating factors and your interleukins. Over here on the side, I've included the term for low white blood cell count, so that condition is called leukocytopenia. You could give examples and causes here. And then also I have leukocytosis, which is when white blood cell counts are elevated, and you could give examples of different conditions that bring about leukocytosis and their causes. As you can see, I can include a lot of information on an 8x10 paper. By you devising your own flowcharts, trying to organize it, and looking at your material, your lecture notes, putting them together, you're going to learn an awful lot of information. So keep in mind, you could still add some additional information to this flowchart. I hope this was helpful.